In this lecture, we are going to continue with enzymes. We had started off with enzymes in the last lecture. We'll continue with that topic and look into enzyme inhibition as a therapeutic module. And the rest of the and the rest of the lecture will be talking. And during the rest of the lecture, we will be talking about lipids and structures of lipids and reactions of lipids. So last time when we talked about enzymes, we were able to see how enzymes are a huge advantage in comparison to uncatalyzed reactions. Enzyme catalyzed reactions are much more faster. In comparison to the uncatalyzed reactions, enzyme catalyzed reactions are much faster, they're highly specific, and they also use very mild temperature and pH. So not only that, the enzymes can also be specifically modulated. You can regulate the activity of an enzyme either by an activator or an inhibitor, and thereby you can actually control some of these reactions much better. The enzymes work as platforms. They have an active site where the substrates can come and bind. And once the substrates bind, the enzyme will change the conformation such that there will be old bonds broken and probably new bonds can be formed. And that leads to the formation of products. Once the products are formed, they can no longer fit in the active site, so they get released, and the enzyme is back in its original shape and conformation so that it can catalyze more reactions. So that is why we need very small amount of the enzyme because they get recycled after each reaction. The way the enzymes work is by reducing the activation energy. If you look at this diagram here where we have plotted energy on the y-axis and the reaction progress on the x-axis, if we have an uncatalyzed reaction, you can see that the uphill is much more steeper, meaning the activation energy is going to be much higher in an uncatalyzed reaction. However, if you look at the enzyme catalyzed reaction, which is this green curve, you can see that it's going to bring down the activation barrier. The energy required to cross the barrier is much lower, and that's why you can see many molecules can now go to the product side. So earlier, probably one or two molecules could go through the energy barrier, but because the energy barrier is much lower, even those molecules which would normally fall off, they're able to cross the barrier and go to the product side. And that's how we are able to have an increased rate or the higher number of molecules being converted into products within a unit time. Another thing to note here is enzymes, all they do is they affect the rate of the reactions. So enzymes only increase the rate of the reaction. They do not, so important to note that they do not affect either the thermodynamics, meaning any of the delta H or delta E of the reaction. These are unchanged. They remain as is. They will be the same whether we have an enzyme catalyzed reaction or an enzyme catalyzed reaction. You will end up having the delta H or delta E that will be the same. It doesn't change. The second thing to note here is they also do not affect the equilibrium. Equilibrium talks about reactant to product conversion. So we have reactant being converted to products. So this is the equilibrium. So the enzymes do not favor the reactants nor the products. If the reactants were favored in an uncatalyzed reaction, enzymes will not change that. So equilibrium meaning KEQ value will not be changed. Now the enzymes are classified based on the type of reactions. So there are 6 classes of enzyme. So there are 6 classes of enzymes, and these are based on, as I said, the type of reactions which you have already seen, the basic types of reactions. The first one here is an oxidoreductase. An oxidoreductase, as the name suggests, is involved in redox reactions. Whenever a molecule is getting oxidized or another molecule is getting reduced, the enzymes that catalyze these reactions are the oxidoreductases. One of the enzymes that you will commonly see in the metabolic pathways, especially glycolysis, so we have multiple dehydrogenases that are involved in the electron transfer. So these are going to be accepting the electrons and giving it to the other compound. The second type of enzymes are called as transferases. Again, as in the name, these are involved in transferring groups between substrates. It can be an acyl transferase, meaning it's going to transfer an acyl group, or aminotransferase, meaning it will transfer an amine group, or phosphotransferases, meaning they're going to transfer phosphate groups. So kinases are an important class of transferases that transfer phosphate group between ATP molecules and substrate. Kinases, these are extremely important because they are present in many different types of regulatory pathways. The next type of reaction is a double displacement. So the transferases, where they catalyze the single replacement type of reactions, where the group gets moved from one to the other. The double replacement will be something like the acid-base or even like hydrolysis reactions. So as in the name again, these are called as hydrolases, where a water molecule is going to break the bond. So we have water as HOH. It will break a molecule and add. This will be the hydrogen here, and the OH will go to the other part. So if you remember when we talked about carbohydrates or even proteins, when they get broken down, polymeric forms get broken down to monomers, that reaction is by hydrolysis, and those reactions in the body are catalyzed by hydrolases. The lipases, amylases, esterases, again, the name suggests what type of compounds are going to be hydrolyzed by those enzymes. So here we see lactonase, phosphogluconolactonase, that is involved in transfer of the phosphate group in the sugar molecules. Then we also have the fourth reaction is a decomposition reaction, meaning you're going to break a bigger molecule into smaller fragments. So you have an AB that gets just chopped off. There is no addition of any other compounds. It's a pure bond breaking between the two compounds. So these enzymes are called as lyases, and lyases, you have seen some of these lyases probably when you so lyases break the bond, and unlike the hydrolases, lyases do not add any compound. When hydrolases break the molecules, they add water to the fragments, but here it's just a bond breaking reaction. The next two types will be isomerases. Isomerases are those that are involved in converting one isomer to another isomer. So we have like glucose, for example. Glucose is converted to fructose. So glucose, both of them have the same molecular formula with the C6H12O6, but one is an aldohexose, the other is a ketohexose. So these two are structural isomers, and the isomerase that converts a glucose to fructose is called as isomerase, phosphoglucoisomerase. The last reaction is a synthesis reaction when we combine two compounds to give rise to a bigger molecule. So these reactions are catalyzed by what are called as ligases. They ligate, meaning they connect two or more molecules together, and thereby they form the chain. So these ligases can be some sort of enzymes which are involved in condensation reactions. And in DNA replication, if you remember, there were the Okazaki fragments, and those Okazaki fragments were connected by a DNA ligase. And all it does is it's going to condense two molecules together, and if it is a dehydration reaction, it is going to be releasing a molecule of water. But sometimes you need not have water molecule being released. It's just pure bond formation between A and B. There. Now the classes of enzymes are very useful because that's how you can analyze what type of a reaction you're looking at whenever you're looking into your metabolic pathway. The enzymes are extremely sensitive to the conditions. So they are active in a specific range of pH, or an optimum pH, or an optimum temperature range. Anytime we go beyond these on either side, if we go higher than the optimum temperature or lower than the optimum temperature, the enzymes will lose their activity, or their activity can get diminished. So here we have shown on the first graph what the effect of temperature. So this particular enzyme is active at 37 degrees Celsius. The maximum activity is around 37 degrees Celsius. So if you go any temperature lower, you can see that the activity drops because we are looking at activity on the y-axis. Or if you increase the temperature on the right, again, there is a drastic drop in the activity of the enzyme. So as long as we maintain very close to the optimum temperature, the enzymes will be active. Anytime it goes on either side, the enzymes get denatured, which means they lose their tertiary structures, and therefore they cannot be active. Now the pH is also very similar. So if you are like plotting the activity of the enzyme on the y-axis against pH, you can see that there is an optimal pH, somewhere around 7 in this case. The enzyme activity is maximum at this pH. And if you go on either side, the enzyme activity drops, again, pretty drastically. And it's very narrow window that we are seeing here. So the enzyme activity drops drastically by the time you reach 1 pH unit on either side, right there. Enzymes are also sensitive to substrate concentration. As you go higher and higher, the excess substrate can limit the enzyme activity. And sometimes we call this as feedback inhibition. So enzymes can get, if there is more of the products, so let's say, for example, we have A being converted to B. So this is catalyzed by an enzyme. If there is, most of the time these reactions will be, so let me show this one. So these reactions are reversible reactions. So let's say there is an enzyme that is catalyzing the conversion of A to B. Now, if there is a very high concentration of B, the product, or if there is a high concentration of the substrate itself, the enzymes can get inhibited. So there is a crucial concentration around which the enzymes will be maximally active. So when working with enzymes or when understanding metabolic reactions, all of these parameters should be considered in order to estimate the enzyme activity under certain conditions. So at this point, I would like to stop here, and I would like you to answer these questions. You can write these answers on a separate sheet of paper, And there are 5 questions in this lecture. You can answer all of them on the same page and upload it on Canvas. So the first question here is, when you boil an egg, what happens to the proteins in the egg? You can pause the video, write it down, and then come back to the video. The second question is, salivary amylase is active in slightly alkaline pH of saliva. So what happens to the enzyme as the saliva with the enzyme enters the stomach? Will it still be active or is it going to be inactive? Think about it and write down the answer. All right, I will continue with the lecture. You can pause the lecture and work on these questions and come back to this part. Now, the enzymes are also dependent on certain coenzymes or cofactors. The coenzymes or cofactors are helper molecules that facilitate certain reactions on the enzyme. For example, if there is a redox reaction, these cofactors or coenzymes can help in transferring the electrons. They can accept the electrons or they can donate the electrons so that the redox reactions can happen. The enzyme itself doesn't have electrons or doesn't use its own electrons. It will use these molecules from the cofactors or coenzymes, and those molecules can donate or accept electrons, thereby helping in the redox process. Generally, an enzyme, most of the enzymes do have some sort of a cofactor or a coenzyme. And these are usually based on the vitamins. That's the reason we need some vitamin, a very small amount of the vitamins, because these vitamins form the coenzyme or the cofactor. And those are really important for the metabolic reactions. Now, an enzyme without the coenzyme, that is called as an apoenzyme. And an apoenzyme can combine with a cofactor or a coenzyme, which usually is a non-protein part. And sometimes it could be just an organic molecule or a metal ion or anything like that. And the coenzyme will be part of the enzyme's active site so that it can facilitate an electron transfer, or it can act as an acid-base donor, like proton donor or a proton acceptor. So whatever the reaction is, many a time these cofactors will be instrumental in helping those reactions get to completion. So an apoenzyme is without the cofactor, but when you combine the two, it is called as a holoenzyme or a whole enzyme. And the whole enzyme is when it is going to be active. The apoenzyme without the coenzyme or the cofactor is not going to be active, even though if it has folded into the right conformation, it still needs that coenzyme or the cofactor in the active site for it to be functional. One of the most common, you can see that we have a lot of different types of coenzymes that are involved in metabolic pathways. Thymine pyrophosphate, TPP. And the source of this is the vitamin, as I mentioned, like thiamine B1 is really important for thymine pyrophosphate. One of the common, the electron acceptor, FAD or NAD, these two are very important in glycolysis, Krebs cycle, or even in the electron transport chain. These are involved in all of these three reactions that is part of the central metabolic pathway. These ones are important to pick up electrons. So FAD can accept electrons. It can accept two electrons and two protons, thereby getting reduced to FADH2. So without this, the redox processes in the citric acid cycle may not go through. And so the deficiency is going to be pretty consequential. So riboflavin, the vitamin that forms the basis of FAD, is riboflavin. Now the NAD is based off of niacin, vitamin B3, pyridoxal phosphate, which is vitamin B6, pyridoxine, biotin, pantothenic acid, folic acid, all of these are going to be acting as so the basic molecules that will be useful for the formation of cofactors. And these cofactors are necessary for most of these enzymes to be active. Now, why do we work so much about enzymes? What is so important about enzymes is we have almost 99% of the reactions that occur in the body are regulated or catalyzed by enzymes. So if there is a disease condition where an enzyme is overactive, when there is a particular metabolic pathway is overactive or you want to stop a particular pathway, then we can design small molecules that can act as inhibitors of the enzyme and thereby shut down the entire pathway. One of the examples that I have here is an enzyme called telomerase. You may have heard of this enzyme. It's an enzyme that adds nucleotides to the ends of the chromosome at the end of every cell division. This is normally the telomerase is not very active in case of normal cells. So you can see that it's going to extend. This is the DNA duplex, and the enzyme has a complementary RNA, and thereby it just keeps on extending the DNA polymer towards the 5' to 3' can keep on getting extended. Now the telomeres are important because they tell when the cells have to go through apoptosis. Normally, most of the normal cells will have certain number of cell divisions, and after which the cells will go through apoptosis or programmed cell death. However, in case of cancer cells, the telomerase is highly active and it keeps on adding the ends so that the DNA replication keeps on going without differentiation, thereby leading to cancer cells. So if we have to target cancer cells specifically and not affect the normal cells, then we know that this enzyme is highly active in case of cancer cells. So if we were to design a small molecule that can inhibit this enzyme, then we can selectively target those cells, like the cancer cells, where these telomerases are highly active. And that is one of the strategies that has been explored in order to design therapeutic interventions for cancer. So we design inhibitor. If you were to design an inhibitor that goes and shuts down the telomerase, then effectively cancer cells will not be able to multiply and the cancers can start to regress because there's no more mass collection. Undifferentiated cells are not going to be constantly synthesized. They're going to get stopped. So you can see that the enzyme, the crucial part of this one is recognizing that there is an enzyme that is active exclusively in cancer cells and trying to come up with small molecules or drug molecules that can specifically bind to these enzymes. That's how we can actually use enzyme inhibition in clinical applications. There are many, most of the, if in the current clinical development, many of the compounds that are being synthesized or evaluated, they target one enzyme or the other in the body. Some of the examples here, for example, HIV protease. HIV protease is a very important target to design small molecules that can inhibit or block viral replication. We also have HMG, the statins. You have heard of statins that are used for cardiovascular, controlling cholesterol. Basically, in cardiovascular disorders, it's used for controlling the cholesterol biosynthesis in the body. All they do is they inhibit a particular enzyme called HMG-CoA reductase, which is the most important crucial enzyme in the synthesis of cholesterol in the body. So when you inhibit the enzyme, then you don't have biosynthesis of cholesterol, thereby the blood cholesterol levels also get lowered, and that's going to help in reducing cardiovascular issues. There are a whole bunch of examples which you can actually look at. As I said, we have cancer that we talked about. There is another kinase, tyrosine kinase inhibitors that can be used for anti-cancer drug treatment, anti-cancer activity. Many of the antibiotics like penicillin, they inhibit the cell wall synthesizing enzymes. And these cell walls are only present in bacteria and not in the animal cells. So we have selectivity right there. Even if we take antibiotics, it's not going to affect our cellular activity. There are also many of these MAO inhibitors. These are used for neurological disorders where they can treat depression. The inhibiting the enzyme will reduce the depression, so thereby it's going to be clinically relevant. So at this point, I have another question. And again, you can pause at this point, answer this question, and come back to the lecture. So we have heard of carbon monoxide poisoning. Whenever there is a fire, the first thing is to ensure that the people are not exposed to carbon monoxide for more than a few minutes. 20 minutes or 30 minutes is the maximum. And the reason the carbon monoxide poisoning becomes fatal, what do you think is the reason? What is it inhibiting? It's acting as an inhibitor. And what do you think it is inhibiting in the body? All right, so you can pause and work on this one, and then you can continue with the next topic here. So the next chapter is lipids. And lipids are also a type of biomolecules, although these are not the traditional polymers that we have seen so far. The carbohydrates and proteins, they have the repeat units exactly the same. Sugar or glucose sort of units keep on getting repeated in a polymeric chain, or amino acids are going to be repeated in a polymeric chain to form those large structures. But in case of lipids, the types of monomers can change. It's not necessarily exactly one type of monomer, but nonetheless, they do have polymerization very similar, and that's why these are also classified as biopolymers. Now, the lipids are defined as organic substances that are relatively insoluble in water, but soluble in organic solvents like chloroform, ether, or benzene. So they're not soluble in water, but they're soluble in nonpolar solvents. Now, lipids are extremely important because they do have multiple roles in the body. First of all, they are used as energy fuels for ATP generation. They store energy. So we can use the hydrolysis of fats to produce ATP molecules when there is deficiency or when there is carbohydrate limitations. There are also structural components of cell membrane, plasma membrane. We'll talk about that, the important components of plasma membrane. And they're also precursors for hormones and vitamins, steroidal hormones. They act as thermal insulators. They're important for the skin, and the skin acting as a physical barrier as well as an insulator. They act as protective. They form the bilipid layer, so they protect the organs as well. Because of the fact that they are non-polar, they also help in the absorption of fat-soluble vitamins. They're important for transporting lipids in the blood. The lipoproteins, we're going to talk about that. Lipoproteins are proteins with lipids that are involved in transporting lipids within the body. And they serve as surfactants, whereby they reduce surface tension, especially cholesterol or these molecules they can form sort of amphipathic, meaning it can have some amount of polarity on one end and predominantly non-polar part on the other end that can act as a surfactant that will reduce. We have these surfactants in our lungs, which is why the lungs don't collapse on itself. They improve, like, taste and palatability. Many a time, this is the reason why fatty foods are going to be more tasty, because they help in the improvement of taste. And in case of neurons, they are extremely important, because the brain neurons, both and throughout the entire nervous system, the neurons are covered with these lipids. Especially, we call them as sphingolipids. The myelin sheath that is present in the neurons is made up of sphingolipids, and they act as insulator, and they also help in the saltatory movement of the signals. Now, lipids are classified into three types: the simple lipids, complex lipid, and derived lipids. So, the simple lipids, as the name suggests, they're simple in that they're esters formed of fatty acids with various alcohols. So, we have an alcohol. Alcohols have the OH group. When you combine that with a fatty, a fatty acid meaning it has a carboxylic acid. So this is going to be an alcohol functional group. And then we have the carboxylic acid here. So when these two get condensed, you can see that the OH of this one and the hydrogen here will be removed as a molecule of water. And then we end up connecting the two to form this sort of a functional group. What is this functional group called as? So this is an ester. So fatty acids, simple lipids are nothing but esters formed when alcohols are condensed with carboxylic acids. And the specific type of alcohol that is very common is the glycerol here. And the fatty acids contain multiple carbon chain. You can see that we have long chain fatty acids. Generally, these are going to be at least C18 all the way to C22 or C24. The number of carbons here can range from anywhere from 18 to 24. So the glycerol is an alcohol in the sense it's a triol. Glycerol has three hydroxyl groups, which is why you can have three fatty acids forming those ester linkages. So you can see this is the glycerol backbone right here in the blue. And when it is condensed with the fatty acids, each of the fatty acids can connect with the OH groups on the glycerol, and thereby you have three fatty acids connected to one glycerol. And this is how it looks like. So you can see the glycerol part here, and the black and white here is going to be the fatty acid chain that you see here. So when glycerol is condensed with fatty acids, it forms triglyceride. Tri meaning three. So three fatty acids linked to one glycerol is a triglyceride. And triglycerides are nothing but these are esters, because they consist of that ester bond between the alcohol and the carboxylic acid. Just to give you a little more idea about this, so we have the glycerol backbone here, and the glycerol has formed the ester linkages right here. All three fatty acids are joined by esters. You can see you can have a saturated, we call this as a saturated fatty acid, when you don't have any double bonds, no double bonds, or pi bonds. And these two here, you can see that they have pi bonds here. There's one here, and there are three double bonds. These are called as unsaturated fatty acids. So we can have a mixture of these, like we can have just one saturated fatty acid, two unsaturated, or any combination of these ones. And that is all dependent on where they are present in the body and what is the functional role it decides. That is that decides what type of fatty acids are present in these triglycerides. As I said, this is a condensation reaction or a dehydration reaction, the synthesis of triglycerides. We have the alcohol here, each of these alcohols here in the glycerol moiety, and we have the OH group from the carboxylic acid group on all of these fatty acids. There are 3 fatty acids that can interact with 3 alcohol groups on the glycerol, and in the process, these guys will be removed as 3 molecules of water. And then there is a connection between the glycerol and the fatty acid and those esters that we see here. Make sure you're able to understand that this is a synthesis reaction, it's a condensation, just like how we had the amino acids condensed together to form peptides, or carbohydrate basic monomeric units condensed together to form glycoside. They're going to form polypeptides, polysaccharides, and the bond in those cases is going to be called as glycosidic bond. Now moving on to complex lipids. Now complex lipids, they contain a fatty acid. So they have an alcohol, fatty acid, and a prosthetic group, meaning an additional group which can be in the form of, it can be a phosphate group when it is called as a phospholipid. It is going to be, so this is going to be a phosphate group here. So if you have a phosphate group, then it is called as phospholipid. If you have a glucose or any sugar, glucose or sugar, then it is called as glyco. The glyco is for sugars. It's called as glycolipid. And then if you have a protein associated with a lipid, that is called as a lipoprotein. So these are complex because they have other biomolecules associated with them. So phospholipids are, you have probably heard of this or come across this in your biology classes. Phospholipids are those that contain a phosphate group, and the phosphate group is part of the alcohol, that glycerol molecule. And you can see that one of the alcoholic groups is being now phosphorylated, so you have a phosphate group, and 2 of the other alcohols are now involved with the fatty acid. So each of these phospholipids have what is called as a polar head. This is a polar head because it contains the phosphate groups, and the phosphate group is going to be negatively charged. So there will be 2 minus actually. Yeah. So we have a polar head, which is the phosphate group right there. So the top part, with the glycerol with the phosphate forms the polar head, and the fatty acid chain will become the nonpolar tail. So you have the alcohol plus the phosphate group, both of which are polar, and that is why the top part becomes polar head. And you have a carbon chain here, just the carbon-hydrogen chain, which is nonpolar, and that's why the tail becomes nonpolar. So you can see that when you have an unsaturated fatty acid versus a saturated fatty acid, there will be a change in those fatty acid chain structures. If you have a saturated one, it just forms a linear, zigzag form, and then it can pack very well. But the moment you have one or more double bonds right there, you have these double bonds. And what type of double bonds are these? Are they cis or trans? So these are cis double bonds, because of which you can see that those 2 are facing the same side of the double bond, and that leads to the kink, meaning there's a bend structure. And this sort of a kink will minimize very tight packing in the membranes. So the unsaturation is important for the fluidity of the membranes. The more double bonded structures that you have, the more fluid the plasma membrane will be. So these are important components of the plasma membrane. We have a bilipid layer. So each of them, so there is a phospholipid, there's one phospholipid here and another phospholipid here. And both of them will be packed together such that their non-polar interiors will be facing each other, and the phosphate groups are going to be facing the exterior, because the exterior is going to be aqueous medium. So if you think of a cell, so let's say this is going to be a cell. So if you think of a cell, so this is aqueous inside and there is aqueous outside. It's all water-based. So the barrier, like whenever you have these, the membrane barriers, then you want to make sure whatever is facing the aqueous part, it should be polar because it should be compatible with the water layer. It's the same thing here. The polar heads will be facing outside because this is also aqueous and that is more compatible. And in between the two is when we have these the non-polar chain, and they form. So because of the non-polarity, they won't allow things to go through. They form a barrier and thereby not only act as protective layers, they also help contain all the material within the cell. The next class of complex lipids is the glycolipids. The glycolipids have very similar structure, like you can think of phospholipid right here. You can see this is a phospholipid, and the phospholipid is attached to a sugar, and these sugar units can be anywhere from 2 to 6 monomeric units that can be joined together, and that forms a glycolipid. The sugar moieties are going to be used as some sort of receptor. So these are extremely useful because they're very specific. So lead to interaction with signal molecules. So for example, if this is these guys can be part of a receptor which interacts with a hormone like insulin or glucagon that comes and sits on the outside. So this could be the part of the receptor. So the insulin recognizes the glycolipid and then anchors on this glycolipid part of it. The glycolipids are very, very common in the neurons. So if you remember, a neuron is going to have an axon and dendron. So you can see the axon is going to be having those myelinated sheaths right there. Now, if you look at the membrane structure of each of these myelinated part, you can see that they have a lot of these glycoproteins on the surface right there. So the membrane is made up of a lot of these glycoproteins that are responsible for picking up any signals and transferring it along the axon terminus. So in terms of the basic structure, so this is what we see in case of CNS. The cerebroside is an example for a glycolipid. It has a backbone called as a sphingosine, and then we have a fatty acid chain, and then a sugar unit. So instead of having a glycerol backbone as we had in case of phospholipids, in these cases we have a different type of alcohol-based backbone, and this is called as sphingosine. It also has some sort of an amide linkage right here. So if you remember, so this is an amide linkage. And what other functional group can you identify here? There's an alcohol here, and there is an ether here. So this is what I would like you to do. Whenever you see these structures, it's a good habit to just identify these functional groups so that you get more comfortable and you're going to be confident on the exam also when such questions are asked as part of a multi question type. Now, finally, we have these lipoproteins. The lipoproteins, the prosthetic group is like, or the additional group is the protein here. So these lipoproteins are extremely important because one, they're useful in digestion. But during digestion, the fats, the lipids are going to be absorbed as chylomicrons, and that is because of the formation of these lipoproteins. And in the blood, the lipids are carried using these lipoproteins. We have very low density lipoprotein, VLDL. We have low density lipoprotein, LDL, and high density lipoprotein, HDL. So you have also read or you're aware that the HDL and the LDL levels are very important parameters to assess the predisposition for any cardiovascular disorders. So the lipoproteins are slightly different because they're much bigger in structure than just the phospholipids or the glycolipids. They consist of these like a whole bunch of triglycerides that are packed with the phospholipid layer right there, singular layer right here. These triglycerides, and the layer has proteins, specific proteins embedded in that phospholipid layer. And they also have some amount of cholesterol, which makes it much more nonpolar. So these fatty acids are also bound to cholesterol, and that is how most of the fatty acids or the triglycerides are going to be encapsulated in the form of a lipoprotein, and this is how it gets transported in the blood. So if you see in the blood, we have RBCs and we have WBCs and all of that stuff. Now we also have HDL and LDL, which are important carrier proteins. They carry the lipids from one part of the body to another, or whether when it is being absorbed from the digestive system and then they have to be transported to the different parts, or when there is breakdown, the adipose tissue has to be broken down and transported for energy purposes. That also is transported using these lipoproteins. So basically, they form some sort of a vesicle with triglycerides being packed inside, and we have a phospholipid layer, and the lipid layer does have proteins, apolipoproteins that are embedded in there, and that is responsible for recognition and they anchor onto the cells and deliver these triglycerides into those cells for energy production. So we have, in terms of clinical relevance, when we have very high, having a higher amount of the high density lipoprotein is a good indicator, because that means we don't have too many degradation products. The low density lipoprotein or very low density lipoprotein, if we have higher amount of this, that means many of these byproducts are floating around, or there's a high amount of triglycerides or cholesterol in the blood that leads to the formation of atherosclerosis. So these guys can end up just getting deposited on the walls of the capillaries. And if there is significant amount of these depositions, then that can cause inflammation. So you can have deposition of these lipid fragments, and we can also have the inflammation plaques that are formed. So once you have the plaques being at the same point, if it starts increasing, then the lumen of the blood vessels will be narrowed down, and that leads to blood. If it is strong enough, then it can block the entire vessel, can lead to cardiac issues. So that is the reason we want to have a higher ratio of HDL to LDL, so that most of the lipids are actually sequestered into these high density lipoproteins, such that they're taken away from the blood and deposited into the cells. We have derived lipids. Derived lipids are those that are obtained by the hydrolysis of some of the lipids. So when we hydrolyze the lipids, we end up getting fatty acids, steroids, cholesterol, and vitamin A or D. These are all smaller fragments of the larger lipids. So these are called as the derived lipids. Finally, I want to highlight the reactions that you need to know about the lipids. So lipids can go through two types of reactions. When you have a fatty acid condense with an alcohol like glycerol, you end up getting the triglyceride, which is an ester. And in the process, we have removed 3 molecules of water, and that is what is shown there. So this is called as the synthesis or the condensation reaction. And if you were to hydrolyze, if you were to break the ester, then we end up going back to glycerol and the 3 fatty acids. So that is hydrolysis reaction. So the forward is condensation, the reverse is going to be hydrolysis. And in this case, water, and you can add a little bit of an acid or a base to enhance the rate of the reaction if this is not done under biological conditions. These sort of reactions happen in the body in presence of enzymes. They catalyze these reactions, so they happen under milder conditions. But if we are doing these reactions on a lab scale, then we need to change the conditions. The last reaction that I want to highlight today is the soaps. So formation. So the triglycerides are the most important raw materials that are used for synthesizing soaps. And soap is nothing but a triglyceride when it is treated with a very strong base. And this is usually called as a lye. So when triglycerides react with a strong base like sodium hydroxide or potassium hydroxide, they end up getting hydrolyzed and forming the corresponding salts, the salt of the acid, salt of fatty acid. So this is called as the salt of the fatty acid is called as the soap. That is what is used as a soap. And because of the fact that it has both polar and non-polar part, so the non-polar part is the R group, whereas the salt side, the ester, the salt of the carboxylate, the fatty acid here, the salt is going to be the polar one. So polar one will dissolve all water-soluble dirt, and non-polar part will dissolve all the oily residues, whether it is in the clothes or whether it is in the dishes. They end up picking up both types of dirt and clean the material. So that is why soaps are very, very useful, because they are amphipathic, meaning they have both polar and non-polar part, and thereby dissolve both types of dirt and clean the materials. So with that, we are done with this lecture. Before we end, I have another couple of questions. Make sure you finish these questions as well. So there are a total of 5 questions on this lecture. We had 3 questions so far, and these are the last 2 questions. And again, I want to make sure that you understand that you have to write these answers on a separate sheet of paper and load it on Canvas. There is a point for each of these questions that will be added towards your final grade. And if you have any questions, feel free to email me. If I do, hopefully I should be able to reply to that within 24 hours. If not, within 2 days, I should be able to reply to these emails. Given it's a holiday weekend, I might end up taking a little longer. But nonetheless, I will definitely respond to these emails. Or you can also come and talk to me in the office hours on the 4th. All right, I'll let you go. You guys have a nice rest of the day, and I'll see you for the next lecture.